In today's episode, we are joined by Professor Sarah Teichmann from the University of Cambridge, a leading figure in computational genomics and single-cell biology. Her work deciphers the cellular heterogeneity of complex tissues through large-scale single-cell transcriptomics, shedding light on the dynamic orchestration of gene regulatory networks in health and disease. How do individual cells navigate developmental trajectories? What can the human cell atlas reveal about the molecular architecture of our organs? Join us as we unravel the cellular code at unprecedented resolution.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Today I'm happy to talk to Sarah Teichmann. Hello, Sarah, and thank you for joining this show.
Hi there. Thank you for having me.
So a question I like to ask every guest to start off the little podcast is, and to ease into the topic, is how did you become interested in biology in the first place and then in pursuing a career in science?
It's a big question. I guess for me, that goes back to school, really. And I was incredibly lucky at my school in Karlsruhe, actually in Germany. I was at the European school there that we had a really wonderful biology and chemistry teacher when we were sort of, 13, 14, 15, 16. And he provided an after-school club, basically for kids to play around with the chemistry lab and also kind of basic molecular biology experiments. And what I started to do was a project investigating the change in the metabolism of leaves in autumn, when they change color and they kind of decrease respiration and the changes in kind of reactions that were catalyzed by leaves in autumn. And that was super fun and was kind of eye-opening in the sense that, as a kid, I then realized there are all these hidden molecules inside our cells. There are all these, biochemical reactions that are happening that are really the essence of life. And that's what got me interested in biochemistry. And eventually I then, from Germany to the UK to study natural sciences at Cambridge University, where I was lucky to have the opportunity to combine different subjects, physics, maths, chemistry, biology. It's a kind of integrated course. And that was really perfect for, what's now bioinformatics, data science, ML, and so on in biology. And that's sort of where we've ended up. Yeah.
So there's a lot of science to be discussed, and I hope I can capture the full picture, and maybe we can work together on doing that. But first things first, you had and still have an incredibly multifaceted career at the intersection of biology and computation, as you just alluded to. But maybe just to give a general picture, and for those unfamiliar, could you introduce yourself and your current role?
Sure. So I'm Sarah Teichmann. I'm located in Cambridge, England, and I'm currently chair in stem cell medicine in the Department of Medicine and the Cambridge Stem Cell Institute in Cambridge, England. I also spend part of my time at GlaxoSmithKline as vice president of translational research and also involved in a couple of startup companies and scientific advisory boards.
So did you leave academia then to be at GSK?
No, I'm super fortunate actually in the sense that the University of Cambridge and GlaxoSmithKline have kind of agreed to allow me to have this joint position, which is somewhat unusual kind of at that level. And but there have been previous Cambridge University professors who've done that before in the past.
So you're
not completely unique in that sense.
Yeah. So you started with biochemistry and evolved toward computational biology. How did that transition happen?
Well, so as I said, so I studied natural sciences at Cambridge, so it's like an integrated course, and I started off with physical sciences, and so that basically meant that I was able to take physics, maths, as well as kind of organic and physical chemistry and so on. And in the end, I did end up studying biochemistry in my last year at Cambridge as an undergrad, and then I actually did a research project in NMR spectroscopy in sort of simulating different labeling patterns of amino acids with different magnetically kind of active atoms, and that got me into sort of scripting and then eventually the sort of programming of biological data sets. And I mean, that's where I really got I sort of caught the bug basically for computational biology, and decided to do a PhD that was sort of theoretical computational with Cyrus Chotia at the MRC Laboratory of Molecular Biology. Now, it was very unusual at the time, and out of about 60 group leaders at the MRC Laboratory of Molecular Biology, Cyrus Chotia was actually the only computational group leader, so it was a very kind of exotic activity. I mean, nowadays, obviously, we're much more familiar with, data-driven approaches in biology and AI/ML, methods, driving biological discoveries and so on. But at that time, it was pretty unusual. But it was a great time because it was, the mid-90s was the time when the first completely sequenced genomes. I mean, they were obviously small bacteria, E. coli and yeast and so on. But there was that richness of sequence data, as well as, of course, the three-dimensional structural data in the protein data bank. And that was a rich playground to play with from a data point of view. And what we did was essentially integrating those two data types, so the proteins of known 3D structure and the sequences, and then project the domains of known structure onto the sequences in order to predict the domain combinations in the proteins of the genomes. And that may sound completely trivial nowadays, but at the time it was a real eye-opener to understand that even short bacterial proteins consist of multiple domains, that they evolve by duplication and recombination, and that they originate from a small number of protein families. And so it was a really fun time kind of for discovery and understanding of the sort of bigger picture of protein evolution, in completely sequenced genomes, and was also good training.
So how did the computational biology change from that time to now?
I mean, it's changed in two ways, I would say. Part of it is driven by new measurement techniques, if you like, and next generation sequencing is probably the biggest one, the single biggest sort of step change, I would say, in generating huge amounts of biological data. But, before that, we had microarrays and so on. I would say the other thing that's changed is the AI/ML methods. So, in those days, we had things like hidden Markov models and support vector machines and so on. But then really the deep learning, I think, really changed things. And now obviously sort of generative AI and agentic AI. So there are two changes. I mean, on the one hand, changes in the biological types of data that are available, and obviously, where we are at the moment with the Human Cell Atlas, we wouldn't be there without single cell and spatial genomics. And so that, it's really the technologies that have really changed things, and then it's the AI/ML methods.
So when you look back at the career path you took until now, were there key moments or key mentors that influenced that career?
Well, my PhD and postdoc mentors are obviously, really, incredibly important to I guess the way I look at science, and what I've learned from them is sort of what I've learned from them is kind of epigenetically imprinted in my approach, I guess. And I've mentioned Cyrus Chotia, who was my PhD mentor in the structural studies division of the MRC Laboratory of Molecular Biology, and then I went on to do a postdoc with Janet Thornton, who was in the Department of Biochemistry at UCL. And both of those are obviously, hugely influential in how I look at science and my career and so on and so forth, especially because, I then went back to LMB to be a faculty member, and, I stayed for about 12 years. And Cyrus was there basically for that time, although at the end, he sort of retired and unfortunately kind of was unwell then after that. And Janet, of course, was also incredibly influential, not just in my kind of early training as a postdoc, but also because I later joined the European Bioinformatics Institute, where she was director, from really one of the incredibly influential director from 2001 to 2000. Sorry, she was the director in kind of a very formative period of EBI, 2001 to 2016, and I was there as a joint faculty between EBI and Sanger from 2013 to 2016. So again, kind of influenced by her. And in fact, I'm looking at the Thornton building right now, which is a new building on the Wellcome Genome campus, that has her name, and that's absolutely fitting for such an influential figure in bioinformatics.
Mm-hmm.
And such a legendary kind of scientist and leader in the community. There are also other people, who've had huge influence on me. I mean, I would say Carol Robinson, who's the inventor of macromolecular mass spectrometry and was a long-term collaborator of mine for over a decade. We did a lot of work together on assembly of protein complexes and stereotypical pathways of assembly, and she's an incredible inspiration and role model and mentor. And, many others over the years. I mean, I was thinking of Yvonne Jones recently, who, worked with on committees. I mean, Christina Orengo, who was at UCL, a professor at UCL, sharing the same space with Janet Thornton. There are many collaborators over the years. My first PhD student, Madan Babu, has, he's now chief data scientist at St. Jude and in the St. Jude leadership team. Definitely had a big influence. And Madan, Cyrus, and I were actually faculty members at the LMB for a period of years. And, and here, when I came over here to the Genome Campus, we founded the Sanger EBI Single Cell Genomics Center, and that whole team of faculty, was really, really influential in the whole evolution of single cell genomics and driving the methods forward. I mean, both locally and globally, actually.
Mm-hmm.
But yeah, there are a lot of people. Obviously, science is it's a journey of collaboration and teamwork. So there are a lot of people who've been important over the years. Yeah.
So you just mentioned the keyword that I want to focus on next, and it's single-cell genomics. So you have been a pioneer in this field. So what excites you most about this field today?
Well, I guess, now we're really in the era of spatial biology, and it's how those single cells, I think to me, it's how they, it's always been how they interact with each other in tissues to form tissues and organs and ultimately kind of determine our physiology as through cell-cell interactions. And the spatial techniques, that are obviously kind of, you couple and integrate them with the single cell and single nuclear data. The spatial techniques are, as I'm sure you're aware, they're really developing at a rapid pace and extremely exciting. I hope that you've captured also that I mentioned I'm a non-executive director of 10x Genomics. So obviously one of the companies that develops the single cell and spatial technologies but that sort of, biology really happens in 3D and 4D, and getting closer to capturing, the human cells and tissues in their 3D context is incredibly exciting because we have such high resolution and such deep molecular data now that it's a really exciting time, I think, in that sense.
Yeah, before we focus on that for a bit, I want to go back to the beginnings of the single-cell genomics era. And in your lab, you started with single-cell RNA-seq. Why was this the first method that you used?
Yeah, so we were studying mouse T cells, in particular sort of T helper cells and their development in the lab at that time. And we were using, next generation sequencing, so RNA sequencing, chip sequencing, and so on, to understand how their development and differentiation is regulated. And of course, what we had to do was always either take a population of cells and then do RNA sequencing on a whole, tens of thousands, hundreds of thousands of cells, or we were able to image them or analyze them by flow cytometry with studying only a few molecules at a time. And so, but it was very clear that both types of techniques were limited in their ability to really resolve the cell states in an unbiased, comprehensive way, right? Because in the bulk sequencing methods, you are averaging over the whole population of cells and then losing the fine-grained cell states and in the sample. And in the imaging and flow cytometry, you're only capturing a limited number of molecules at a time in a single cell with single molecule FISH, what we were using at the time, and then FACS. And so we were, when Azim Surani and his group, Fuchu Tang, published their Nature Methods paper on RNA sequencing of 4 or 5 cells in the early mouse embryo, it was really clear to me that this was, a real step change and incredibly exciting, with a huge potential for being scaled up to understand, more complex populations of cells that you could then analyze kind of in an unbiased way in terms of using, all the computational methods to then trace the cell activation, cell proliferation, cell differentiation, and so on. So it was more that, the process of naive CD4 T cells differentiating to the different subtypes that we were interested in, which is relatively simple, compared to the complexity of a whole tissue sample. But nevertheless, the existing techniques weren't really capable of describing all the molecular processes that were happening in these cells at the same time. And so when that paper came out, was, to me, it was clear that this was a game changer. But actually implementing those methods in the lab, was super tricky. And Azim, they handpicked individual cells, manually, and then they made the cDNA and amplified and made libraries and sequenced them. And as I said, for 4 or 5 cells, and we tried to use plate-based methods. It's super tricky to actually FACS sort individual cells into wells, and then the amount of material was so low that it would just evaporate unless you were extremely careful. So the protocols were just super tricky. And then, but there were protocols that were developed both by groups in Sweden, by groups in Israel and the US and so on. But it was all very, very sort of, even if it was plate-based or robot, it's very tricky to implement. And then there were companies like Fluidigm, that provided chips, microfluidic chips, which then came along, and they were imperfect, but that was basically where we started making progress. And we actually worked with Azim's lab, who was test driving those Fluidigm chips, and sort of started generating data with them on the mouse T cells. And then eventually, also got plate-based protocols to work with SmartSeq, from Richard Sandberg's lab. And then eventually, of course, then the 10x kind of microfluidic robots came, the droplet-based microfluidics came. And so that sort of evolution of technologies really, empowered the whole field of single cell genomics. I think it's all the different, and it's continuing now, even with all the different companies providing different varieties of protocols, and the multi-omics, with RNA and open chromatin profiling and protein panels, antibody panels with RNA and open chromatin, and EDJ sequencing of B and T cell receptors. So it's, all these technology developments are just really fantastic for biology and biomedical research.
It's interesting, or it was not scripted that you mentioned Azim Surani, because he was the guest in the last podcast episode. So that's
really?
a nice coincidence.
That's funny. So did he talk about that history kind of at all?
Not so much. Not so much.
No, okay. Yeah.
Yeah. We talked about other things. So this single cell work of yours then, I guess, also led to the founding of the Human Cell Atlas in 2017. You are a co-founder of this project. So how did the idea come about and how did you get involved in this?
Yeah, so as I said, we were sort of using, studying mouse immunology with the technologies. And then in 2013, in February 2013, I moved on to the Genome Campus here in Hingston, just outside Cambridge, UK. And I was the first joint faculty between EBI and Sanger, actually. And as I moved, I was sort of negotiating the move in 2012. I was actually on maternity leave with my second daughter, Laura. What we did with 5 faculty across the 2 institutes was write a proposal and get funding for the Sanger EBI Single Cell Genomics Center. And so that was Wolf Reich, Thierry Faut, John Marioni, Chris Ponting, myself, and then a colleague called Harold Swedlow from Sanger, from the sort of platform teams. And so what we did was brainstorming about, what could you do with single cell technologies, both experimentally and computationally, and what biological questions could you address. And during that time, I was discussing the move out to the Genome Campus with Janet Thornton, whom I've mentioned earlier, and also Mike Stratton, who was the director at Sanger at the time. And of course, the Sanger Institute is famous for sequencing just under half of the human genome sequence, the first kind of reference sequence. And it was clear to me that the uniqueness of this campus was basically, the ability to essentially generate large data resources for the biomedical research community, like the Human Genome Project. And with single cell genomics, I mean, the obvious kind of equivalent sort of project, basically, or thing would be sequencing, all the cells in the human body, right? And that was, of course, slightly ridiculous, to think about at the time, but nevertheless, that would kind of, the sequencing all the cells in the human body would sort of be in a way, the equivalent of sequencing the human genome for sequencing the human genome, and for single cell genomics, sequencing the profile of all the cells in the human body. And we started brainstorming, with those colleagues around what would be the simplest thing to sequence. Would you start with a worm? Would you start with a teratoma? There were all kinds of ideas. And for, would you sequence mouse samples, mouse immune samples, or whatever? And in the end, for a whole number of reasons, actually, human tissue samples are actually quite suitable from an experimental and practical point of view for these technologies, because the cells are quite big relative to something like a worm or fly. Many tissues, you can actually disaggregate the cells. It turns out, I mean, the fly has worms, for instance, are really hard in that way because they have a shell kind of surrounding them, and so on. And so we actually ended up then, thinking about essentially human cell atlasing at that time, and then, working on the first sort of human project we ended up working on was actually the first trimester placenta, so which is a super interesting organ, sort of at the very origin of our life at the beginning of pregnancy. But yeah, I mean, the first step was really that we got the funding and the support from both Sanger and EBI with that group to drive forwards the methods and the technology development. And that was really the framework. And then in 2014, Aviv, had pitched to NHGRI on a project called the Human Cell Atlas, so that name, with the same sort of concept of sequencing all the cells in the human genome. And then, there were also other centers in the world that were sort of thinking around this. Clearly, the Swedish technology developers were super important, and, in California, there's a community and so on. So this idea was kind of bubbling up. But then when I guess the next sort of important step was that when in 2016, I started the job as head of one of the departments at Sanger, the cellular genetics. And, that was kind of crucial because it sort of opened up a funding opportunity. And when I knew that I had that job, I reached out to Aviv and called her and said, would you be interested in launching an international consortium together? And we tried to get more funding for this because she hadn't, the NHGRI hadn't sort of bidden. And obviously, this is a project that is clearly hugely ambitious, huge effort, a lot of work, requires a lot of scientists, a lot of funding, a lot of expertise. And of course, we still haven't completed the human cell atlas today. It's still ongoing. So it was clearly the case that it was ambitious. And Aviv replied immediately and we started talking. We had phone calls every week and we organized the first sort of step towards this, which was, a meeting of about 100 people in London in October 2016. So it's 2016, actually, not 2017. And we also had a small kind of pre-meeting in the summer in the US with about 10 or 12 people. And that really, that's what really got the ball rolling. Because at the kickoff meeting in October 2016, there was a lot of enthusiasm, a lot of support from the scientific community, a lot of support from funders. And we formed our organizing committee, which is kind of the coordinating governance body of the consortium, and that's still in place today. And Aviv and I still co-chair that. So that was really how that started. And yeah, I think we would have never dreamed that it would be so successful and so impactful and so influential. And so we're really delighted and pleased and energized about pushing this forwards into the spatial era and keeping the momentum going until we've actually fully completed the human cell atlas.
So what were the biggest scientific and logistical challenges in the beginning to setting all this up and then coordinating this global project?
Where should I start? So I guess both Aviv and I were really lucky to be in a position where we had really amazing colleagues that supported from a logistical point of view. So Aviv had her Klarman Cell Observatory at the Broad Institute, and the staff members there essentially started supporting the human cells. And over at Sanger, we had the cellular genetics program and essentially recruited staff and who all kind of contributed to really setting up the human cell atlas, in the early days. And last night I just met Mike Stubbington, who was a postdoc of mine who then really went on to contribute to driving the whole effort forwards, and Orit Rosenblatt-Rosen who was at the Broad Institute at the time. So it was, from a logistical point of view, it was a huge amount of work, and it was a team effort, I think, internationally, really, to make it happen. From that point of view. But of course, we also had, a global community. And I mentioned this organizing committee, and there were maybe, 20, 25 people who got together, at that meeting in London then and there. And that includes people, from all over the world, from Europe, Germany, from, Asia, the US, Canada, and so on. And since then, we've really broadened our global reach to Latin America, Africa, Middle East, and you might know that we are now a UNESCO partner project. So that's very exciting and, a huge honor to have that partnership with the United Nations Education, Science and Culture Organization. And very few science projects, are supported in that way by UNESCO or big kind of, science consortia. And we have the support of that organization, which essentially has representation in every country in the world. And you might know UNESCO from sort of World Heritage Sites, but it also has that science component. And it supported the Human Genome Project and Human Genetic Variation studies, and we're really delighted that we are partners with them for the Human Cell Atlas community. They like basically the way we champion open data sharing. We are aligned around science diplomacy, as this global effort internationally, and also around the education component. So they also emphasize education and also, making sure that there's inclusive education and science also for women and girls and so on. So it's a really, really exciting time for Human Cell Atlas.
Mm. So when you look back, what would you say has been the most surprising scientific finding from the Human Cell Atlas so far?
There are a lot of, really incredible.
Sorry to interrupt, but are you reading all the papers that come out?
Well, we have to at least review the ones that are part of the Human Cell Atlas publication collection. So if you go to humancellatlas.org, we have a list of publications that are kind of, how can I say, that are sort of endorsed and as part of the Human Cell Atlas collection, and they have to comply with things like, posting of the manuscript online openly, data sharing principles, et cetera, et cetera. So, we do review all of those, and we have a publications committee that does that I'm part of.
Okay.
And so that collection is online and searchable. And then, of course, we also have our publication bundles that are also, reviewed by the committee. And you may have seen the bundle that just came out in November that had 46 papers across Nature and Nature Publishing Group journals, which was really, very exciting and lots of incredible discoveries in that. And it would be hard to pick out, individual discoveries and highlights. But I can pick out a few if you want me to. I guess, one of the most maybe surprising things to some people is just the sheer complexity and diversity of cell types and cell states, right? I mean, if you look at Wikipedia, there are about 200 cells that are mentioned. One of my postdocs recently defined over 180 different cell states in the human T cell compartment alone. Mm-hmm. So that's T cells, T cells alone. I mean, imagine that. And that's, just one subtype of our immune system. And of course, that includes kind of states that are responding to viruses and so on. It includes memory states and naive states. It includes different proliferation states and so on. So it's really a fine-grained classification, but nevertheless, it kind of shows the sheer complexity, that was unappreciated before. And in T cells, I mean, some of the things that we've discovered are innate-like T cells, particularly in human development. So a lot of very unexpected discoveries have been made in looking at the developmental stages during pregnancy, so the human embryonic and fetal stages. And of course, we've been focusing a lot on the immune system and those innate-like T cells and innate B cells, B1 cells, and their roles and where they develop also in different tissues has been quite unexpected and surprising. So you'd expect to see them developing in lymphoid tissues, but we see some developmental states in unexpected places like the skin and the gut. So that's really, really shocking kind of in some sense, and tells us that the signaling molecules that are sending signals to the progenitors, can actually be provided by unexpected cell types. Mm-hmm. So there are those discoveries. There are incredible discoveries in the brain. Kind of the sheer complexity of cells in the human brain again has been amazing. And the Brain NIH Brain Initiative had a publication bundle that's coupled with the brain bionetwork of the Human Cell Atlas community. And in the mouse, there was mouse and human and non-human primate. Actually, in the mouse, there were 5,000 cell types that were kind of clustered in an unbiased way. Again, kind of suggesting the sheer unappreciated complexity of our cells is just beyond anything imaginable. I guess a lot of really important and impactful insights have also been in disease, but even in COVID, the human cell atlas community mapped the viral entry factors in the different tissues, and what that pinpointed was unexpected cells in the eyes, the nose, the oral cavity, where the virus could enter. And that was immediately kind of picked up, from a public health point of view for personal protective equipment, for mask wearing and so on, as support for the public health policies. And that's the reason I highlight that is that that's not even studying of diseased tissue, but it's studying the healthy human cell atlas data and, giving us predictions about where pathogens are entering our body and how we can protect ourselves. And of course, immediately it kind of, gives you the concept of using the human cell atlas as a Google Maps of the human bodies, where we can trace pathogen entry factors, where we can trace genetic variants, mutations associated with rare disease, common mutations associated with common complex disease risk, tracing drug targets and predicting side effects. So in other words, using the human cell atlas from a safety and toxicity point of view. There are all these ways in which that Google Maps at such high resolution and such deep molecular breadth is incredibly powerful and impactful from the get-go, kind of for human biomedical research or biomedical research in general. So I think I hope that paints a kind of picture.
It does. It does.
Yeah.
So your lab has also contributed heavily to the human cell atlas. You focused mainly on the immune system and heart, if I'm not incorrect. And I guess you just talked about it a little bit, what you contributed to this. Can you maybe talk about your efforts here, what you maybe did not talk about just now?
Sure. I mean, so I've mentioned a bit about the immune system, and the placenta was kind of motivated also by, our curiosity to understand how immune tolerance is set up at the maternal-fetal interface. And, the paper by Ventotormo and Efremova et al. In 2018 was a nice kind of exemplar of that, because we were able to sample all the different tissues in the placenta and cover all the different cell compartments. So by cell compartments, I mean, both the trophoblast cells, which are the cells of embryonic origin, the placenta, as well as the vascular cells, the blood vessels and the immune cells on the embryonic side, but then also the maternal counterpart that's from the side of the uterus. And, getting all those tissues in such a comprehensive way was possible through the Human Developmental Biology resource, which is a very precious bioresource here in the UK. And what was very exciting was our ability to trace the receptor ligand interactions across the cells and then understand at the molecular level what are the mechanisms for cell-to-cell communication that are set up that allow that tolerogenic environment to be established at the maternal-fetal interface for the placenta to kind of implant and integrate with the uterus. And that's so important for pregnancy success. And yet it's kind of counterintuitive from an immune point of view, because, of course, the embryonic cells harbor the paternal antigens, which the maternal immune system would naturally kind of reject. But there's a special circuitry of the cells and of the receptor ligand interactions that means that there's tolerance, and there's also the remodeling of the vasculature on the maternal side that allows the oxygen and nutrients to be delivered to the placenta. And the cell atlas of the placenta that we stitch together, and reconstructing the cells and their kind of tissue microenvironments was really an exciting adventure in shedding more light onto those mechanisms. And so that's, that was a great project. And that you're right that we've also kind of traveled into the human heart, which is another kind of vital organ. So the placenta is, set up at the very beginning of our development, and then the heart develops very early as well in pregnancy, and then obviously is crucial to our, our to life, and in beats at a steady pace, really, from early in development all the way to until we take our last breath at the end of our life. And that understanding the cells in the 4 chambers of the heart, and understanding how the beating is coordinated by the sinoatrial node through the cardiac conduction system that, signals to the 4 chambers to beat in a coordinated way, is really one of the other, nice kind of exemplar projects, I think, of our human cell atlas work. And again, both of those projects are international collaborations, with absolutely incredible teams in Germany, in the US, and the UK, which has been just a fantastic, experience.
Mm-hmm.
And I guess in the heart, really discovering the single cell profile of the human pacemaker cells was, a real highlight, kind of especially in the pandemic when it was so hard to work and to get tissues, because transplantation was absolutely, at a minimum in the UK. And of course, these are precious tissues from deceased transplant donors where the organ is rejected for transplantation. And getting, seeing the profile of those pacemaker cells that are spontaneously firing, in from 2 heart samples in the first instance, there were only about 50 nuclei. And we started to become more and more certain that we had discovered the pacemaker cells in human, and, kind of caught the profile for the first time. I mean, that was incredibly exciting also because of all the new insights from the transcriptomics of the pacemaker cells, where we found that they have this very unique glutamatergic signaling with glial cells that kind of hug them and nurture them in the sinoatrial node. And they also have sort of unexpected endocrine signaling, like hormonal regulation by the GLP-1 receptor, which is the agonist of GLP-1 receptor, and that's of course the target of liraglutide and semaglutide. So those are Wegovy and Ozempic, the drugs that are used for weight management.
I see. Mm-hmm.
And so that was, very unexpected. And we stumbled on that through our drug to cell framework for mapping the targets of drugs at the single cell level. So it was really, also assembling the human cell atlas kind of effort, sort of putting together, in a sense, the muscle of the heart with the brain of the heart, the cardiac conduction system, with the endocrine system, which is, hormones that signal at long distances between our organs, and coordinate our physiology. And it was a kind of nice project where putting many of those components together. So that was a lot of fun.
Mm-hmm. I see. So in the interest of time, we have to speed up a little bit because we are reaching the end of your availability. So how do you see the human cell atlas evolving over the next 5 to 10 years? And for how long is this project planned?
So the kind of really exciting phase that we're in right now is assembling data suspension cell and nuclear data sets to release reference data objects for human tissues, organs, and systems. So we have a project where there are 12 postdocs that are funded by the Trans-Zuckerberg Initiative that are working together with our 18 biological networks that represent the different organs and systems, the major organs and systems in the body. And what we're doing is releasing reference data objects on our portal at data.humancellatlas.org. And so we're kind of in that assembly phase now, at least for the suspension cell data sets. And in terms of data generation, we're in a phase, I mentioned we're in the spatial era, so we're in a phase where we're generating and pushing forwards the spatial mapping of the tissues and organs in the 18 bionetworks of the Human Cell Atlas, and also expanding our kind of profiling of samples from humans all over the world. So expanding the kind of diversity of the data sets that we have, and that's facilitated through our global community and our regional networks across the world. And an example of that is the recent paper of the AIDA, the Asian Immune Diversity Atlas, AIDA, by Shyam Prabhakar, Jay Shin, and colleagues in Thailand and India, and Partha Majumdar and colleagues in India and others. And so there's basically the sort of spatial axis, but also the human diversity axis, where there's active data generation. And I guess what we'd love to have is to kind of push that forward and finish the project kind of in the next phase, sort of moving into that new era. And I'll say 5 years, but I was over optimistic, I think, at the beginning, and I expected to kind of be done and dusted within a decade. But then COVID came. I think there are a lot of unexpected sort of twists and turns in the road. But it's also an opportunity now because the technologies have obviously advanced massively, both in the single cell and spatial genomics, but also in the AI/ML, and so that's kind of good in that sense.
Yeah. So for the last 50 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I don't think so, Stefan. I think we've covered a lot of ground.
Yeah.
There's nothing I can think of right now.
That's good. So thank you, Sarah, for your time and for being on the show.
Thank you so much.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.